Hello everyone. So in this session we are going to study about the test cross, monohybrid test cross. The monohybrid test cross, it comes under the inheritance of one gene. So in the inheritance of one gene we studied about the one character, how one character inherit from one generation to other generation. So in the last session we studied about the monohybrid cross, how the tall and the dwarf plant, when we cross these plant, what will be the offsprings, either it is tall or it is dwarf. And in the F2 generation, when we cross the F2 generation offsprings, in the F2 generation offsprings, what will be the ratio of the offspring. So all those things we learned in the last session. So in this session I'm going to describe you, I'm going to teach you about the test cross, how to test the parents of the monohybrid cross. Whatever we have taken in the F1 generation, I told you in the F1 generation we have selected only pure lines, pure breeds, that is homozygous dominant and homozygous recessive. If we doesn't know which parent, with the genotype of parent, in case of some, if we need to cross any parent like in any organism, if we doesn't know either it is in a homozygous condition or it is in a heterozygous condition, in that case we can test the genotype of the parental generation. How to test that? So we'll see that one by one. Test cross. Which test cross it is? It is a monohybrid test cross. Okay, so let me tell you one more time here. The monohybrid cross, what we did here, we have selected the parents. So what I am teaching you here, what I am writing here, I am just writing the schematical representation of monohybrid cross. From this I will come to the test cross. Okay, if I directly jump onto the test cross, I am just refreshing the monohybrid schematic representation. What we have selected in the parent while describing the monohybrid cross, one plant is tall and one plant is dwarf in nature, and just consider that as a phenotype. Okay, so the phenotype is, let me write once again this one, tall, this is the tall plant and this is the dwarf plant. The phenotype is tall and the phenotype of the one more parent is dwarf. And though we said the parents are pure breeds, pure breed in sense, for the tall phenotype we have to write the homozygous condition of the tall, for the tall is capital T. Pure breed in sense there is no recessive gene, there will be no recessive gene in the homozygous condition. So these are the pure breed. And for the dwarf, this is also pure, as I told you, it should come small t. Capital T represent the dominant nature, small t represent the recessive nature or recessive trait. If we take the gametes of these T and T, when we cross these gametes in sense male and female gamete, when these two gametes fertilizes, when the fertilization takes place in the male and female gamete, the next offspring will be capital T and small t. On that day I wrote like this, right? Okay, no problem. There were 4 offsprings, right? So here we got the 4 offsprings. 4 offsprings or 2 offsprings or many offsprings, it depends on the number of gametes. But for the experiment or for the explanation, only 2 or 4 offsprings are more than enough. Okay, so these offsprings are F1 generation offsprings and it is in a heterozygous condition. Alright, heterozygous in sense here one dominant trait is there, one recessive trait is there. When we self-pollinate or when we selfing, when we start selfing these offsprings, selfing means I am taking this offspring and this offsprings and I am considering this offspring as male parent and this as female parent. What will be the resultant offsprings here? So you have to select among these as one male parent and one more is female parent. Capital T, when it split the gametes, when the genes split, those genes carried by the gametes and each gamete having the 50% here, 50% of the gene will go to some of the gametes and 50% whatever the gamete genes are there in the in the pair of genes, those will split. And how it will split? When the gametes when the gametes like it produces, how it produces how the gametes produces by means of meiosis, right? In the meiosis the reductional division takes place. When the reductional division takes place through the core chromosomes, the genes also passes. By that these genes carried by each and every gametes. Here capital T and small t, capital T and small t. Just cross it, capital T and small t, capital T. So here capital T, small t. Homozygous dominant, heterozygous dominant, homozygous recessive nature. By this we got the ratio of phenotypic ratio. The phenotypic ratio is 3.1. Phenotypic ratio in sense the physical appearance of the plant. Out of 4, the physical appearance of the plant or the phenotypic ratio or phenotype of the plant is 3. 3 are tall and only 1 is dwarf. So how we used to write PR or phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. I hope you have understood how to write the genotypic ratio. Take this as 1, like write all the possible offsprings here. Whatever in the checkerboard, just write it once you write it, and this is also there in the checkerboard. Write that also, capital T, and then small t. Just count it. How many capital T's are there? 1, right? How many capital T, small t's are there? 2, right? How many small t's are there? 1. So what I have written here, so this is the genotypic ratio of monohybrid cross. So these are all the procedure we studied in the last session, and considering this, okay, so I'll consider both of the parent, either it is in a homozygous or heterozygous. We are going to test the parents, like for example I'll write the parents here. Which parent we are selecting, we don't know whether it is in a whether it is a pure breed or heterozygous dominant. Okay, so we'll take both the possibilities here. What we are learning here, how to test the parents, either it is in a homozygous condition or it is in a heterozygous condition. How to test the genotype of the parent. For the testing, what we are selecting, both the possibilities. The possibilities will be purebred and the possibilities. This is the possibility number 1 and this is the possibility number 2. In the physical appearance, these two parents appear tall in nature. To this, so this is the possibility number 1. Once I finish this one, I will go to this possibility. Let me complete this session first. So what we have taken here, we don't know which parent it is, in which condition it is. Just I am selecting one of the parent. Okay, by appearing we cannot say the parent, the organism or the plant is in homozygous or heterozygous condition. Once here I have written the possibility. To this we are crossing a recessive, pure recessive one, pure homozygous recessive character parent. Alright, so what will be the next offsprings, what will be the next process. So if this is a parent, the gametes will be, what will be the gamete, capital T and capital T, and small t and small t. Just cross it, just put the Punnett square and capital T, small t and small t. So this is the possibility number 1. When we cross the parent having homozygous dominant to the pure recessive one, the gametes will be like this. Alright, and to the gametes, one of the parent gamete taking on the top of the square and one of the parent gametes taking on the left side. When we start crossing these two gametes, the offsprings will be like this, capital T, small t, capital T, small t. If in the offsprings or in the F1 generation, if all the offsprings are tall, the stem of the height of the stem is tall, or the top plant, all the offsprings plant is tall, so it is confirmed that we have selected a pure line, a pure breed. If the offsprings are tall in nature, the parent is in pure condition, pure line. So when we cross, we will take the possibility number 2. We have chosen the parent, we don't know the genotype of the parent. So the possibility number 2, it is in a heterozygous condition. When we cross this heterozygous condition to the pure recessive one or homozygous recessive, the gametes will be like this. So these are the gametes and this is the phenotype. How you write the parents here? The parents will be heterozygous dominant, heterozygous tall, more than enough, heterozygous tall, homozygous recessive. You have to write like this. Okay, make it correct here also. So consider this as a phenotype and the parents are homozygous tall and homozygous dwarf. Alright, so this is the possibility number 1 and this is the possibility number 2. What is the use of testing the parents? From this we will come to know what we have selected the parents. So if it is in a homozygous condition or it is in a heterozygous condition, if it is in a homozygous condition, the offsprings, what we have got the offsprings, all will be in a tall condition or the stem of the height will be tall, and if it is a heterozygous tall plant, we will see the next thing. Put the Punnett square, take the two gametes on the top, consider this as a male and this will be this as a female right here. capital T, small t, small t, small t, capital T, small t, small t, small t. Look at the offsprings. 50% of the offsprings are tall, 50% of the offsprings are dwarf. Put the ratio, will come to know. Phenotypic ratio of the monohybrid test crosses, possibility number 1. What we are saying here, possibility number 1 and possibility number 2. I am writing the possibility number 2 phenotype now. Phenotypic ratio, phenotype ratio, tall is to dwarf. How many tall plants are there? 2 tall plants. This is tall and this is dwarf. This is tall and this is dwarf. Okay, how many tall plants are there? 1 and 2, right. How many dwarf plants are there? 2. 2 is to 2. 50% of the offsprings are tall plant, 50% of the offsprings are dwarf plants. Coming to the genotypic ratio of the possibility number 2, genotype is, how to write the genotype of the ratios, genotype of the offsprings, right here, capital T, small t. I have written here, no, just write it, capital T, small t. How many capital T, small t's are there? 2 capital T, small t's are there, right here, is to, how many small t's are there? 2. And the genotypes are also 50%. 50% of the genotype of the parent is capital T, small t. 50% of the genotype is small t, small t. So this is how we have to consider the parents, either it is in a homo or it is in a hetero. So coming to the I am writing here the possibility number 1 phenotypic ratio. Phenotypic ratio of the possibility number 1, phenotype, tall, 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 tall. Tall is to dwarf. How many tall plants? 4 tall, 0. 0 dwarf. Genotype, capital T, capital T, small t, that's it, no. So it is also 4 is to 0. So you have to write the ratios of the offsprings as follows. So we have done the test cross of the monohybrid cross. Only we have completed the monohybrid test cross. Alright. So with this, we will move on to the laws. LAWS, laws. What laws? By learning the principles of inheritance and variation, the Mendel, what he found out the rules of principles of variation, principles of inheritance and variation. He made some rules. So this is how. So in the rules, what will be there? So those rules told, or those rules explains how to cross the plants. How will be the genotype of the parent? How will be the phenotype of the parent? When the dominant character express, when the recessive character express, whatever he explained about the rules in his experiment, so that becomes the laws of inheritance. Laws of inheritance. The first law is law of dominance, and the second law is law of segregation. Law of dominance and law of segregation. So law of dominance, in sense, we have learned, we have come across the dominant character, or we have learned about the dominant trait or dominant nature of the organisms. when in what condition the dominant nature expresses. So that is only you have to learn in the laws of dominance and laws of segregation. So that about that we'll discuss in the next session. Before that, I would like to tell you about the test. So we are going to conduct a test for 50 mark. The chapters will be, the biology chapters are chapter number 2 and chapter number 3. Chapter number 2 is sexual reproduction in flowering plants, and chapter number 3 is human reproduction. Right. So the question paper pattern will be like 1 mark question, 10 questions will be there, and 2 mark 5 questions will be there, 3 mark 5 questions will be there, and 5 mark 3 questions will be there. Choices will be there, and come out with that test, and work hard. If you have any doubts regarding to today's session or yesterday or the day before yesterday, don't hesitate to message me and make it clear. And if you have any doubts in the chapter number 2 and chapter number 3, just message me. I'll repeat once again. Thank you.